On a warm night in the forests of Moorea, French Polynesia, a small team of scientists crouches in the leaf litter with UV torches, sweeping the undergrowth centimeter by centimeter. They are searching for snails. Not ordinary snails, snails smaller than a fingernail, each one released here years earlier and marked with a tiny dot of paint that stays invisible in daylight and only catches the light when a torch beam hits it directly. Every marked snail they find on a leaf tonight is proof of something quietly extraordinary, that a captive population is still clinging on in a forest it was removed from decades ago. Then one member of the team leans in closer to a single leaf and stops moving completely. This snail has no dot, no mark of any kind, anywhere on its shell, which means nobody released it here. It was born in this exact valley, to parents that no human being carried in by hand. And that one unmarked animal, sitting perfectly still on a leaf in the dark, is about to answer a question conservationists had been asking for 40 years without an answer. Of the roughly 70 species of Partula tree snail formally assessed by conservationists today, 32 are gone completely, wiped from the planet with no population left anywhere on Earth, wild or otherwise. 10 more exist nowhere except inside a small number of zoos, thousands of miles from the only islands their species had ever called home. Scientists have a specific classification for animals in that position: not endangered, not even critically endangered, extinct in the wild. A label that means, in plain terms, this species has no future left unless something changes, because there is no longer anywhere on the planet where it can survive on its own. On the Society Islands alone, at least 51 Partula species once lived, and today as many as 34 of them are believed extinct outright. Of the 17 still known to exist in any form, 10 survive only behind glass. Ask yourself how that happens. How does an entire genus of animal, spread across dozens of separate, thriving island populations, collapse this completely, this fast? And ask yourself something else. If a species can vanish from the wild that quickly, how long should it take for one to come back? To understand it, you have to go back to a set of islands most people will never visit, and most people have never heard produced one of the most complete real-time case studies of evolution ever documented. The Society Islands of French Polynesia, Tahiti, and Moorea chief among them, are cut into steep, narrow valleys, each one isolated from its neighbor by ridgelines the snails themselves could never cross. A Partula population living on one slope could be visibly and genetically distinct from a population living in the very next valley, separated by nothing more than a few kilometers of rainforest canopy. Biologists going back to the early 20th century used these snails as a living laboratory, tracking how isolated populations drifted apart generation by generation, valley by valley, in a natural experiment nobody had to design. It was slow, patient, unglamorous science. Decades spent counting shell color morphs and recording tiny genetic shifts that most of the public would never hear about. The tradition went back even further than that, to naturalist Henry Crampton, who began publishing detailed studies on the variation and distribution of Partula snails in the 1920s, decades before anyone had reason to think of these animals as anything other than a curiosity for academic study. Nobody running that research in the 1960s and 70s believed they were also quietly assembling the only surviving record of species that were, without anyone realizing it yet, only a few years from disappearing entirely. The shelves of preserved specimens and the notebooks of measurements were meant to describe evolution in progress. Instead, without anyone intending it, they were becoming the last complete portrait of a fauna about to be erased. The disappearance did not begin with the snails at all. It began with a mistake made in the name of correcting an earlier mistake. In the years before the 1970s, the giant African land snail had been introduced to French Polynesia as a food source. It escaped captivity, as it almost always does wherever it is introduced, and it began stripping village gardens and agricultural plots across the islands, growing to the size of a human fist and reproducing without a single natural predator to hold it back. Officials needed a solution, and in 1974 they believed they had found one. The rosy wolf snail, a carnivorous predator native to the southern United States, was released onto Tahiti specifically to hunt down and control the giant African land snail. It reached Moorea 3 years later in 1977 and spread across the remaining Society Islands through the 1980s and into the 1990s. The rosy wolf snail did not do the job it was brought in to do. It is faster than the giant African land snail, more agile, and devastatingly effective at tracking prey by scent trail across leaves, bark, and open soil. But instead of hunting the pest it was released to control, it turned almost entirely toward a food source that was smaller, slower, and defenseless in every way that mattered. The native Partula tree snails had evolved for millions of years on islands with nothing like this hunting them. No chemical defense, no shell thick enough to matter, no behavior built for evading a predator that could follow a mucus trail through the dark as easily as through daylight. Within a single generation, the wolf snail had swept through the lowland forests. Within a decade, it had climbed into valleys scientists had assumed were too remote, too steep, too high in elevation for it to ever reach. Picture it happening in real time. A valley checked one year still holding 6 or 7 distinct Partula species clinging to leaves and bark. The same valley checked 18 months later, and the wolf snails are already there, and the native snails are already gone. Island by island, valley by valley, populations that had taken hundreds of thousands of years to diverge into separate species were being erased in the space of a few seasons. Across the Society Islands, 61 endemic Partula species existed before the invasion began. Within roughly 15 years, that number had collapsed to just 5 surviving in the wild, and even those 5 were confined to a handful of cool, high-altitude forest refuges the predator had not yet managed to climb into. Conservationists still describe it as one of the fastest, most complete extinction events ever driven by a single introduced species anywhere on the planet. Here is the part of this story that almost never gets told in full. The scientists who had spent decades studying Partula purely for evolutionary research were, without ever intending to be, sitting on the only insurance policy these species had left. Small breeding colonies, kept in laboratories in the United Kingdom for genetics work that had nothing to do with conservation and everything to do with academic curiosity, suddenly became the last living representatives of species that no longer existed anywhere in nature. Brian Clark, James Murray, and Michael Johnson, the researchers whose original work had built those colonies, became the accidental founders of what would eventually grow into a full international rescue operation, though none of them set out to run one. One of those captive populations, tended for years by Vivian Frame, is now directly credited with saving the species Partula tahiviana from complete extinction. Without her sustained care of a population that existed purely to support laboratory genetics, that species would simply not exist today, in a zoo or anywhere else on Earth. While the labs quietly held on to the last individuals of certain species, one man spent nearly 2 decades racing the wolf snail across the islands in real time. Trevor Coote spent almost 20 years doing something close to impossible, moving from valley to valley and island to island, physically collecting the last living individuals of populations before the advancing predator front arrived to finish them for good. Imagine that job for a moment. Hiking into a steep, humid valley, not knowing whether the population you are there to save has 3 more years left, or 3 more weeks. Every population Coote reached in time became a captive lineage that would otherwise never have existed at all. Every population he reached, too late, became one more name quietly added to an extinction list. Nobody was publicly tracking at the time. This was not conservation planning done from behind a desk. This was fieldwork run, against a deadline nobody could see coming, racing a predator that never slowed down and never once lost the scent trail. By the early 1990s, the situation had grown urgent enough that an informal scattered rescue effort turned into a coordinated international program. The last remaining individuals of 15 Partula species and subspecies were gathered and brought into managed captivity, formalized under a program led by the Zoological Society of London and a growing coalition of zoos spanning 2 continents. Edinburgh, Bristol, Detroit, Marwell, Poznan, Riga, St. Louis, Sedgwick County, Whipsnade, Woodland Park, Wuppertal, and Zoo Schwerin were joined over the following years by institutions including Akron, Ardis, Chester, Dusseldorf, Disney's Animal Kingdom, and Jersey, each one agreeing to take on responsibility for maintaining a fragment of a species that no longer existed anywhere else on the planet. St. Louis Zoo would go on to coordinate the American side of the effort, while London Zoo coordinated the international and European side. In practice, this was a living gene bank scattered across 2 continents, with more than a dozen separate institutions, each quietly responsible for a piece of a story that had already ended in the wild. What followed next was not a quick turnaround. It was decades of nothing dramatic happening at all, which is exactly the part of a conservation story that rarely makes it into any documentary. Partula snails lived out generation after generation entirely inside tanks and terrariums, breeding under carefully managed conditions, their entire genetic future dependent on keepers tracking lineage by hand to avoid the inbreeding that comes from populations this small and this isolated from one another. There was no leaf litter to break down, no forest canopy overhead, no predator to avoid, and for years, no clear timeline for whether any of them would ever see their native islands again. The rosy wolf snail was still out there across most of the Society Islands, uncontrolled, and releasing captive snails into a landscape still crawling with the exact predator that had wiped them out the first time would have accomplished nothing except restaging the same extinction on a smaller, sadder scale. So, for most of the 1990s and the 2000s, patience was the entire strategy. Keep the lineages alive. Keep the genetic diversity intact, across zoos that had never seen each other's collections in person. Wait for a door that had not opened yet. Picture what that patience actually looked like from the inside of an invertebrate house on a wet morning in a city thousands of miles from any Pacific island. A keeper checking humidity levels in a row of tanks before the public even arrives. Logging which individual bred with which, by hand, to keep track of a family tree small enough that a single careless pairing could undo years of careful management. Misting leaves. Adjusting temperature by fractions of a degree. None of it filmed, none of it dramatic, none of it likely to be noticed by a visitor walking past a small tank of snails on their way to see something larger and louder. This is what most of conservation actually looks like. Not a single dramatic rescue, but thousands of small, repetitive, unglamorous days, stacked one after another for over 30 years, done by people who had no guarantee any of it would ever matter to anything beyond the tank in front of them. The turning point came only once conservationists and the French Polynesian government, working through the country's Environment Directorate, identified specific release sites where the predator population could be closely monitored and actively managed, giving captive-bred snails an actual chance at survival rather than a second death sentence. Reintroductions began in earnest during the 2010s, and by the 2020s they had become an annual event, with snails flown roughly 10,000 miles from zoos across the United Kingdom and the United States back to the forests of Moorea and Tahiti. Picture that journey for a moment, a creature smaller than a fingernail, born in a climate-controlled tank, in a city it will never see the outside of, packed carefully into a small ventilated container, alongside dozens of others from the same lineage, checked in as cargo, and flown across an ocean and a continent before finally being carried on foot up a forest trail to a specific coordinate marked years in advance. On release day, teams from multiple zoos and the French Polynesian Environment Directorate walk the same narrow forest paths together, each carrying containers of a species some of them have spent their entire careers breeding but have never once seen outside a tank, in a forest that is not the one it evolved in, but is, at last, a real forest, with real rain and real predators to avoid, rather than a sheet of glass. Every snail is placed by hand onto a specific leaf, in a specific microhabitat chosen for humidity and shade, and then the team moves on to the next coordinate, and the next, sometimes placing hundreds of individuals across a single afternoon before the light fails and they have to climb back down. Every snail released carries that same tiny dot of paint, invisible by daylight, catching torchlight at night, letting researchers walking the same release sites months and years later tell at a glance which animals were placed there by human hands and which, if any at all, were not. In 2023 alone, more than 5,000 snails were released across Tahiti and Moorea. The following year, that number passed 6,000, spanning 10 different species and subspecies bred across the international zoo network, including more than 3,000 individuals of the species Partula nodosa shipped in from American zoos through St. Louis alone, roughly 1,000 of which had been raised at St. Louis itself. Then the monitoring began, turning up something the program had been waiting decades to confirm. Population checks in the release zones, carried out month after month by teams walking the same narrow transects with the same torches, started finding healthy numbers of the critically endangered species Partula taeniata, surviving well past the point where earlier, smaller attempts had failed. It was the first hard proof that captive-bred snails, after 2 full human generations spent entirely away from wild conditions, could not only survive back in native forest, but establish themselves there for good. It was not yet the final answer the whole program had been built around, but it was the strongest sign yet that the answer was finally close. Which brings the story back to that single unmarked snail found sitting on a leaf in 2024. That snail was Partula taeniata, the very species Vivian Frame's captive colony had rescued from the edge of extinction decades earlier in a laboratory in the United Kingdom. Finding an individual with no paint mark meant only one possible thing. It had hatched in the wild, to parents that were themselves reintroduced, in a forest that had not produced a single wild-born Partula taeniata in roughly 40 years. Following that discovery, the International Union for Conservation of Nature formally reclassified the species, moving it entirely off the extinct in the wild list and reclassifying it instead as critically endangered, a status that still carries real risk, but represents something the species had not had in 4 decades. A genuine wild population, capable of reproducing entirely on its own. In the months after that first discovery, researchers found additional unmarked individuals living outside the original release area altogether, meaning the population was not simply surviving in place, it was beginning to spread. A species can be erased so completely from its native habitat that an entire human generation is born, grows up, and grows old having never known it existed anywhere, except behind glass in a handful of zoos most of them will never visit, and it can still come back, provided the people responsible for it are willing to wait as long as it takes without giving up on the lineage. This was never really a story about saving one small mollusk most people had never heard of. It is a working demonstration that extinct in the wild, a classification that sounds and is usually treated as functionally permanent, does not have to be the end of the sentence. The threat that caused all of this in the first place has not gone anywhere. The rosy wolf snail is still present across most of the Society Islands, uncontrolled outside the small number of managed release zones where reintroduction is currently possible at all. Of the 10 Partula species and subspecies that still exist only in captivity today, most are still waiting for a safe place to go home to. Decades into lives lived entirely in tanks, passed down from one generation of keepers to the next. Every reintroduction remains a calculated risk, dependent on predator control holding firm at a specific site, on genetic diversity surviving across captive generations bred far from any ancestral forest, and on funding and staff time continuing to be available for a program most people outside conservation circles have never once heard of. The success on that one slope of Moorea is real, and it took 40 years to arrive. It does not undo the fact that the rest of the genus is standing almost exactly where it stood 4 decades ago, extinct in the wild, waiting for its own version of that one unmarked snail. For every species like Partula taeniata that gets a headline moment on a single leaf in a single forest, there are still several more sitting in tanks in Europe and North America with no confirmed release date at all, their entire future still resting on the same kind of quiet, unglamorous care that kept taeniata alive for 4 decades, before anyone outside the program ever heard its name. Partula tree snails were never just a curiosity for evolutionary biologists to admire under a microscope. In the forests where they still survive, they spend their lives moving slowly across leaves and bark, feeding on decaying plant matter and fungal growth, cycling nutrients back into the soil beneath them, and helping keep disease from spreading unchecked through the leaf litter. Multiply one snail's slow, patient grazing across a leaf by tens of thousands of individuals working the same square kilometer of forest floor for generations, and what looks like an unremarkable animal becomes a genuine piece of forest infrastructure, quietly maintaining a balance that nobody notices until it disappears. Losing them was never only the loss of a shell pattern that took biologists decades to document and classify, one that filled museum drawers and academic papers with detail found almost nowhere else in evolutionary biology. It was the quiet removal of a small, specific job the forest floor had relied on for far longer than anyone alive had been around to notice was even happening in the first place. Back on that same slope in Moorea, under the same UV torchlight, the research team logs the unmarked snail's exact location, photographs it precisely where it was found, and leaves it undisturbed on its leaf. Nobody planted this animal here. Nobody carried it across 10,000 miles inside a climate-controlled container. It exists because a scientist in a laboratory decades ago kept a population alive for a study that had nothing to do with saving anything. Because one field researcher spent 20 years outrunning a predator he could never fully stop, and because a scattered international network of zoos agreed to hold on to a species that had already, on paper, ceased to exist. It went extinct in the wild for 40 years. Nobody told it to stop being a snail. If species patient enough to wait 40 years for a way back home are the kind of story that pulls you in, we have put together a full, detailed deep dive on the other Partula species still waiting in captivity right now, and what it would actually take to bring each and every one of them home, too.